Autoimmune gastritis is a chronic inflammatory condition that primarily affects the gastric mucosa, driven by an autoimmune response against the parietal cells of the stomach. This multifaceted disorder has gained increasing recognition for its intricate pathogenesis, distinct morphological features, and potential complications. In this video, we delve into the realms of autoimmune gastritis, exploring its pathogenesis, morphology, and associated complications. Pathogenesis. Autoimmune gastritis is characterized by an immune-mediated attack on the parietal cells of the gastric mucosa, resulting in the destruction of these essential cells. Central to its pathogenesis is the production of autoantibodies, particularly antiparietal cell antibodies, APCA, and anti-intrinsic factor antibodies, IFA. These antibodies target key proteins involved in gastric acid secretion, such as the H+/K+ ATPase and intrinsic factor. The autoimmune response is orchestrated by a T-cell-mediated process involving both CD4+ and CD8+ T cells. Genetic predisposition, particularly the association with HLA class II genes, plays a role in susceptibility to autoimmune gastritis. Environmental factors, such as infections and exposure to certain toxins, may trigger or exacerbate the immune response in genetically predisposed individuals. Morphology. 1. Parietal cell loss. Autoimmune gastritis is characterized by the progressive loss of parietal cells, leading to reduced gastric acid secretion. The destruction of parietal cells is often accompanied by infiltration of the gastric mucosa by lymphocytes and other immune cells. 2. Chronic inflammation. Chronic inflammatory changes, including lymphocytic and plasma cell infiltration, are common findings. These changes may extend into the submucosa and deeper layers of the gastric wall. 3. Metaplastic changes. Atrophic changes in the gastric glands can lead to metaplasia, particularly intestinal metaplasia, which is a precursor to gastric adenocarcinoma. Associated complications. 1. Vitamin B12 deficiency. Autoimmune gastritis can result in impaired secretion of intrinsic factor, leading to malabsorption of vitamin B12. Vitamin B12 deficiency may manifest as megaloblastic anemia and neurological complications. 2. Pernicious anemia. The loss of parietal cells and intrinsic factor can lead to pernicious anemia, a type of megaloblastic anemia. Early detection and treatment of vitamin B12 deficiency are crucial in preventing pernicious anemia. 3. Gastric adenocarcinoma. Long-standing autoimmune gastritis, especially in the presence of metaplastic changes, is associated with an increased risk of gastric adenocarcinoma. Regular surveillance and endoscopic examinations are recommended for early detection of neoplastic changes. 4. Neurological manifestations. Vitamin B12 deficiency associated with autoimmune gastritis can lead to neurological complications, including peripheral neuropathy and cognitive impairment. In conclusion, autoimmune gastritis unfolds as a complex interplay between genetic susceptibility, environmental triggers, and immune-mediated destruction of parietal cells. The morphological changes encompass parietal cell loss, chronic inflammation, and metaplastic alterations. The potential complications, ranging from vitamin deficiencies to an increased risk of gastric adenocarcinoma, underscore the importance of early diagnosis and comprehensive management. As our understanding of autoimmune gastritis evolves, so too does the promise of targeted therapeutic interventions aimed at modulating the autoimmune response and mitigating the associated complications.